Well, good day to everyone joining us and welcome to today's Xtalks webinar. Today's talk is entitled Oral Dosage Innovation for Pediatric Drug Delivery. My name is Ryan Muse and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank AustinPx, who developed the content for this presentation. AustinPx Pharmaceutics and Manufacturing is a contract development and manufacturing organization providing analytical and formulation development services and cGMP manufacturing for small molecule drugs. AustinPx specializes in phase-appropriate development strategies, speed to clinic and market strategies, and bioavailability enhancement of poorly soluble molecules, including their next-generation amorphous dispersion platform, Kinetisol technology. Now I would like to introduce our speakers for today's event. Dr. Dave Miller currently serves as the Chief Scientific Officer at AustinPx, where he leads the pharmaceutics and analytical development teams and oversees the application of the Kinetisol technology. He specializes in formulation and processing technologies for improving the oral bioavailability of insoluble small molecules. He has applied his expertise toward advancing numerous drug candidates through all stages of development, from early discovery to line extensions. Dr. Zachary Warnken serves currently as the Director of Pharmaceutical Development at Via Therapeutics, LLC, a small pharmaceutical startup focused on delivering innovative pharmaceutical solutions. His work focuses on developing vital new treatments for underserved patient populations, including inhaled biologics and small molecules, as well as the devices that deliver the medication. But now, without further ado, I'd like to go ahead and hand the presentation over to our first speaker for today, Dr. Zachary Warnken. You can begin when you're ready.
Perfect, thank you, Ryan. If I can, yeah, there we go. Excellent, thanks. Yes, I'm very excited about this. Today we're going to be talking about a drug that I've worked with for many years. Unfortunately, fortunately, as we'll learn, at Via Therapeutics, we're fantastic at taking ideas and turning them into possibilities, but as we'll learn a lot through this webinar, it takes more than just possibilities. So Austin Pharmaceutics was able to chime in and take that possibility and kind of turn it into a product. We'll learn why that was so hard. So the starting point for this, which was funded by a contract with NIAID, was to develop a second-line treatment for tuberculosis in the pediatric population. So tuberculosis is fairly widespread globally, including in children, affecting 1.2 million children, and that was just in 2002 numbers. And it's growing in resistance, so the need for these second-line treatments is already fairly prevalent, but it's growing year after year. Additionally, clofazimine isn't just used for tuberculosis, even though that was the main focus due to the contract, but it's also used for non-tuberculosis mycobacterial infections, which are on the rise, even as we are better and better at targeting TB. But the drug clofazimine that was already in the market as an approved product, lamprey, which originally was approved for the treatment of leprosy, is not ideal for the treatment of pediatric patients for a few different reasons. One, it's discontinued in the US. So even though we don't have a lot of TB here, we do have NTM infections. So having something in the US would be great. Lamprey can be obtained in other countries for the treatment of TB, but it comes as a soft gelatin, lipid-filled lipid matrix with micronized drug in it. So while children, are growing and there are a bunch of different weights, even during the time in which they're having to undergo treatment for TB, splitting these soft gelatin capsules with the lipid matrix really isn't feasible. And so what they do now is they'll just double up the doses on one day, and then you just wait a couple days and then take the next dose, instead of having a low dose given every day. And that has its own drawbacks as well. One, it's not as consistent, so compliance could be an issue, although with the clinics that are often dosing this, they can overcome that. The other issue is you're getting really high Cmaxes in that case to be able to accommodate the dosage form that's on the market. And there are issues with doing so. This drug can accumulate in the tissues. One of its side effects is literally staining of your skin while taking this drug. So you can actually see color changes in your skin from accumulation of this drug. So that only will get worse with higher concentrations. And more seriously is GI impaction, which would result in discontinuation of clofazimine. So being able to have lower concentrations more consistently would be better, and we could do that if we could break the dosage form down. There are some other issues though. So this drug is very red, which is why it can even stain your skin, and so we need to keep that in mind as we're going to be dosing pediatrics. So while making a straightforward suspension with clofazimine would be fantastic, there's many drawbacks to that approach as well. While we'd be able to just change the volume to be able to address these different weights, we'd be directly adding the drug into the mouth, which could cause staining of the teeth of these patients. We're not really sure how long the staining, if it was like of your teeth, would last, and so that could be detrimental to the psyche of these patients. And additionally, there are other considerations we have to think about when we're shipping liquids. Most of the TB cases we're going to be treating with this drug are going to be in low and middle income countries. So being able to have a very cost-effective, easy to ship dosage form is going to be needed to be able to address this patient population. And so a solid oral dosage form would be most preferred. So we got into testing clofazimine because we were actually working on another contract of micronizing the clofazimine for direct delivery into the lung. This way we could get higher concentrations only in the lung and maybe expand the indication as well and improve its efficacy. During the control studies for some of those arms, we dosed the drug orally as well. And we found with our micronized drug products that were micronized to a size that were really intended to be inhaled, we were also overcoming some of the bioavailability limitations and actually being able to treat TB in a mouse model. So this was really promising when we saw that there is a need for additional oral drug products as well. And so that's why we decided that let's go ahead and utilize that same product and micronized clofazimine that we've been working on and we know is able to provide the pharmacokinetics that we need as an oral solid dosage form for pediatrics. This would enable just a more straightforward bioequivalence approval pathway as opposed to the inhalation product, which also may have difficulties in the pediatric population anyhow. So to work on this pediatric dosage form, we started with understanding what the dose requirements were going to be. That lamprey product comes as a 50 milligram and 100 milligram dosage form. So we needed to figure out where we're going to fill the gap for all the patients who need less than that. And typically the dose is about 1 milligram to 2.5 milligrams per kilogram. And so we need something that's going to go from that 50 milligram and less. Additionally, we wanted it to be readily swallowable by patients. And so keeping that small mini tab size. Mini tabs in some studies have been shown to be more palatable to young children than even suspensions, thanks to being able to mask taste and make it easily and very fast to swallow instead of sticking onto the tongue. But we wanted it to be able to be easily countable, and we'll look at this a little bit more detail in just a second, but for both patients and potentially in clinics, as this drug is sometimes dosed with direct observational therapy to ensure that patients are taking their full doses. So as such, and due to some constraints with the device, we wanted it in a 2 millimeter mini tablet. And so that meant we needed 2.5 milligrams per mini tab. And so to do this, kind of provides one of our first constraints. We need 50% of our micronized hydrophobic drug per mini tab to be able to meet this. And additionally, we want to be able to add a coating on top of that so that it's not disintegrating in the mouth, but will then dissolve and disintegrate after the patient has swallowed the dosage form. So first, just looking at kind of what that device looks like, this is by Philips Medisize and it is available for purchase. This is kind of what we envisioned would be fantastic for use with our mini tabs, as you can easily adjust the number of mini tabs that need to be dosed based off the weight of a patient. It is able to be contained within a child-safe container that still protects from humidity and light, just like any HDPE bottle. And so it's also, while the device component is maybe not the cheapest aspect of this, it's a very small component and it still goes on to regular bottles. So we're keeping costs down, which is important for the patient population we'll be targeting and the countries in which those patients reside. We're able to then count these little mini tablets, which in the next picture, I think you'll really be able to appreciate how small these are. So being able to, you could easily swallow 20 of these on a teaspoon, but being able to count 20 of them out by hand is very difficult. So that can kind of be overcome here. We can easily adjust based off weight, and this is relatively easy and straightforward to use, which is great, without compromising on stability. So here, these are actually the coated mini tabs in the upper right picture that we'll discuss that were designed and developed at AustinPx, but you can see by just how small these little 2 millimeters are, and that comes with a lot of considerations we have to think about while designing this type of dosage form. So being able to fill a 2 millimeter dye is not trivial, especially if your powder isn't fantastic at flowing. And as you may be able to appreciate, having a very hydrophobic, micronized down to 3 micron size does not create a highly flowable powder. And then when you need 50% of that powder to be that very poor flowing material, you run into some design constraints that have to be overcome. Additionally, we wanted to be able to add that coating so that it's not dissolving in the mouth. That requires certain strength of those tablets so they're not have low friability so that they can go through the fluid bed process and get coated. And additionally, these need to be able to scale up. So while there are tips that are able to punch multiple mini tabs in a single compression, which really helps being able to knock out a lot of these at once, that small diameter, that low fill creates a lot of variability. We need to be able to do this from not just small scale, but be able to scale up if this is going to be a product in the future. And any small milligram difference per mini tab is a very large % difference in the final dose and weight of the mini tab. And so the reason this is so challenging is because of clofazimine and its physicochemical properties. It's very, very lipophilic. When people say brick dust for talking about a hydrophobic drug, clofazimine is the prime example because it literally looks like red clay. It was originally used as a dye before it was a product for pharmaceutical use. And so it has a very large food effect, which is probably can't be overcome because it's so lipophilic. And we have now micronized it into a very, very small size. Remember, initially with the thought that it would be inhalable. And so we're less than a D90 of 3.5 microns. And we wanted to keep that because we knew that provided us the bioavailability advantage that we were hoping would then be bioequivalent to the product already on the market, and that we knew was able to treat TB in a mouse model. So that powder is not easy to work with. Dave is going to go through more exactly how we gather this data, but initially looking at the rheometry data, I mean, we have very low bulk density. we have very high compressibility, but we have poor flowability. No surprise. So like I said, we have high compressibility, but we don't have very good tabletability. So as we increase those pressures, we don't consistently get an increase in tensile strength, which we really need to be able to have hard enough tablets to be able to go through this fluid bed coating process later on. And so, so because of the constraints we have to be able to meet this patient population and where they are, and the difficulties of clofazimine, we start out with having a requirement for high drug load with this very poor tabletable, likely to have friability issue drug powder that's micronized of clofazimine that has poor, while great compressibility, poor compactability. It doesn't create these strong tablets. Again, we have strict size constraints that we need. We need those 2 millimeters, especially if we want it to be able to function with that device, that counting device that is already on the market or available for marketing. And then we need it to be able to go through the fluid bed coating process without breaking the mini tablets to get that functional coating. And so, like any good sponsor of any project, we just take all those problems and hand them off to Dave at AustinPx to deal with.
Thanks, Zach. All right, let me go ahead and take over my screen. So yeah, that's a good transition. So you know, it's a really common scenario for us at Austin Pharmaceutics, where you've got a very innovative company like Via Therapeutics, and Zach and his team have recognized an unmet therapeutic need in the marketplace. They've developed a very innovative approach to addressing that need, but recognizing the technical challenges that they face in going from concept to commercialization. They need a CDMO partner that's more than just sort of transactional services, but actually someone who will be an extension of their team, who has tools and experience and expertise to take that challenging concept and move it forward, interacting with the client at every step, identifying issues, and proposing solutions to the client to resolve all the problems that can come up in the course of development, tech transfer, and manufacturing. And I think this webinar represents just a really great example of how we interacted with Via Therapeutics on a very challenging formulation problem, and worked with them in step to solve to problems, to slay all the dragons that come up along the way, so that we can take this product to patients. And I think, as Zach laid out, the problem statement from a patient's perspective is one that's is very challenging and one that needs to be addressed with the number of children that are infected with these mycobacterial infections every year. So with that, I'll dive into to my portion of the presentation. So right up top, we'll talk about the essential tools that were required to do data-driven development and taking the concept and the formulation and the target product profile that Zach and the team at Via came up with, and converting that into a robust product and manufacturing process that we can take through clinical development and all the way toward commercialization and getting this mini tablet product to the patient. So the tools that were essential for us in being successful in this project are shown on the slide. The Style 1 Nano compaction simulator has a wide breadth of capability that allows one to evaluate compression with various ways of modifying the compression profile to simulate commercial tablet production. And given that we're making a very difficult mini tablet formulation, this tool is absolutely essential to identifying potential issues and red flags early on a material sparing basis, projecting those out to larger scale manufacturing, and making the corrections that need to be made to ensure robust processing very early in development. So as a compaction simulation tool, the Style 1 is excellent for what we were asked to do in taking a formulation from small scale and project it out towards commercialization. And then in step with the compaction simulator is using the FT4 powder rheometer to analyze powder properties in multiple different ways. So again, we can address potential flow issues, segregation issues, all of the problems that come up with powder handling and compression. We can evaluate those using the FT4, and it's absolutely essential in this case because there's as Zach said, very little margin for error when it comes to a mini tablet. All of the usual issues in compaction of tablets are magnified when you go to a small geometry, a 2mm mini tablet. So we've got to dial everything in to a much greater extent than you would for a normal monolithic tablet. So let's start with what our initial approach. So Zach and the team at VIA did an excellent job at sorting through the infinite formulation permutations that one can go through in developing a mini tablet with a high drug load of a highly compressible API. So as Zach already pointed out, you got this 50% of micronized clofazimine that's highly compressible, meaning you don't have to apply much pressure to collapse all the porosity out of the compact. And for an insoluble drug, that porosity is essential to achieving disintegration, dissolution, therapeutic efficacy. So that constraint, high drug load of a poorly soluble compound that has high compressibility properties, is a huge challenge. So Zach and the team really did a great job of evaluating a number of different commercial tablet blend excipients, combining those with disintegrants, lubricants, screening them out based on disintegration time and the visualization of capping. So this slide represents a small piece of the total formulation evaluation experimentation that VIA did at a small scale to arrive at that last row, which is the formulation that we transferred in and began our direct compression evaluation. So it's 50% drug load with that's largely the external phase comprising Prosolve as a binder, Actisol as a disintegrant, SSF as a lubricant, and SLFs have some wettability properties as well as enhancing lubrication. So when we took the formulation in, right away we used the FT4 powder rheology tester to evaluate the blend to identify any potential issues with direct compression. So you can see that the analysis looking at bulk density, the basic flow energy of the powder, stability index, flow rate index, specific energy of this directly compressible blend. Based on the outcome of the FT4, we noted potential issues with the low bulk density being only just under 0.25, and also powder cohesion risks that the powder is changing under shear and over time. So these are all sort of red flags that we were able to communicate very early using the FT4 powder rheometer. We went ahead with the direct compression evaluation using the style 1 to see if those red flags do actually transition into compression issues. So very simple process. We do blending and compression using the style 1 and a 9-tip, I think initially looking at a 2.5mm mini tab tooling, 9 small tips per tool. We've got a little video here. Okay. Yeah, for those who haven't seen the style 1 in operation, that's what it looks like. And as you can see, there's a lot of different ways that you can modify the movement of the parts to simulate different aspects of high-speed tableting. So it's an excellent compression simulation tool to evaluate at a small scale powder properties, but also project out towards large-scale tablet production. So the results for our direct compression trials, where we saw that the tablet weight was below target and variable, as well hardness and thickness of the tablets naturally were also variable. As you see in the photo, there was quite a bit of punch sticking observed with the API, and as a result of poor flow, poor dye fill, we saw a lot of capping, and just in general a lot of variability, a lot of challenges in powder handling, being that it's 50% of this difficult to handle active ingredient. So the conclusions were that based on these direct compression trials, we needed to improve flow, increase the flow of the blend so that it can more consistently flow into the small die of the mini tab tooling, and as well as optimize lubricant levels to minimize adhesion and reduce the ejection force. And we also would evaluate the method of addition for blending. So with that, we made a second attempt, taking that same formulation and converting it from direct compression to dry granulation. The formulation that Zach and the team worked out previously was ideal for this approach. So we took the formulation, put all the ProSolve and the SLS into the intragranular phase, added some lubricant to facilitate dry granulation, did the compaction on the Style 1 Nano for making the slugs, as I'll show you in a minute, and then post-slugging and after milling, we add additional disintegrant and lubricant to the granules in the compression of the mini tablets, and then finally conclude with the functional coat. So here's just the process flow diagram. So again, blending the micronized API with ProSolve and SLS, slugging on the Style 1 Nano with 15 millimeter round flat face tooling, milling in a conical mill, externally blending the dry granules with lubricant and the disintegrant, and compress in the Style 1 Nano. So evaluating the dry granulation, first looking at the slugs. Again, the dry granulation formulation was suitable in terms of slugging, good hardness, tensile strength on the slugs. When we milled those down and compared the flow properties of the dry granules alone and in the final tablet blend compared to the direct compression blend, we saw significant improvements in bulk density naturally. Bulk density increased by almost twofold, and the flowability energy also increased, and the powder became less dependent on flow rate and time. So the properties were more static. So a lot of positive improvements moving to dry granulation. Then when we took these dry granules, a couple of different formulations, and compressed mini tabs using the Style 1 Nano, we noted that punch sticking and capping was resolved. So we improved the powder handling significantly by increasing bulk density, increasing particle size distribution through dry granulation. And when the flow into the small die cavities of the mini tab tooling was much improved, which obviously increases the likelihood of being on target in terms of weight and hardness. So overall weight variability was low, hardness variability was, as I'll talk about in a second, was fairly consistent, although we saw two populations. So in certain situations when we were compressing where the weight was spot on, we saw great tablets, acceptable hardness, but in other situations, the weight was slightly below target and we saw low hardness. So there was still it just kind of highlights the issue of you don't have to be off by much with a mini tab to be below the acceptable limits. So just this point is meant to say that with mini tableting, all the problems of typical tableting are amplified significantly. So again, even though we made significant improvements through dry granulation, there was still work to be done. That said, with dry granulation, all the tablets that were tested that met the requirements disintegrated very quickly. So that was a win. But when we went to dissolution analysis on tablets made at varying compression pressures, as we do compression analysis and compression profiling and do dissolution as a function of pressure, we saw that the compression force dependence on drug release was unacceptable, highly dependent on the compression force, and thus we needed to once again pivot our approach. So in the third attempt was looking at wet granulation. And so once again, the current formulation that we were working with was deemed to be highly suitable for direct transition to high shear wet granulation. We developed aqueous binder solutions with different binders at a 10% level. And the potential advantages over dry granulation were many. One, there was elimination of pre-granulation powder handling. Again, the API, as Zach described, is very difficult to handle in terms of the staining potential, the flow, the static. So being able to just combine the materials into the granulation pot significantly improves the handling of the powder itself. Again, improving blend and blend homogeneity by producing bound granules with binder in an aqueous form, improve the particle size distribution and thus the overall blend uniformity. And then these granules should have more favorable properties for compression, not only achieving higher bulk density and better flow, but also a bit more porosity. That'll improve compactability, which resulting in better tablet properties, including friability, and ultimately scalability of the blending and tableting process is expected to be much improved, utilizing wet granulation to bind up this fine drug powder with the external phase to create granules that are better flowing and better compressible for mini tablets. So just an overview of the wet granulation process flowchart. It's again, high shear wet granulation using the micronized drug, SLS, and MCC, and binding those up with by high shear granulation with a binder solution of 10% binder. And we looked at HPMC, PVP binders, as I'll show you in a moment. Then after screening the wet mass and tray drying, there's a conical mill step to mill these materials through a 1 millimeter screen and recycle. Then finally, the wet granulated granules are, after drying, blended with disintegrant and lubricant to form the final tablet blend. And then once again, did the compression on the Style 1 Nano with a 12-tip 2 millimeter tablet tooling with varying the compression profile to fully evaluate the compressibility of the powder and project out towards large-scale tablet manufacturing. So we look at the characterization of the wet granulation blend compared to the dry granulation blend with different permutations of wet granules. We saw what we expected, that the wet granulation produced higher bulk density with similar tap density to dry granulation. Saw overall improved flow, larger particle sizes, in some cases more fines. We found that all three binder solutions, the copovidone, povidone, and HPMC, produced acceptable granules, and that those granules were all very comparable in their properties. So using an aqueous binder at 10% was very effective, almost independent of the type of binder. So then when we took this wet granulated tablet blend onto the Style 1 Nano, we saw a significant improvement in the stability of tableting run. There were no fill height or force adjustments were needed, very stable. There was excellent weight uniformity for all three formulations, which indicate very good blend flow. So once again, powder flow into these very small 2 millimeter dies has to be excellent in order to have a robust formulation and process. And so we were able to achieve that with wet granulation. Between the different types of binders, there was some variability in compression that required us to dial in the compression parameters according to formulation. But that's what the beauty of the Style 1 Nano. You can fine adjust the press to hit your targets very readily, and we were able to do that for all three. What was interesting was the disintegration times were found to be longer with the wet granulated blends versus the dry granules. So more on that in just a moment. And but friability was very low, which obviously is important in this case because this terminates by applying a functional coating and a fluid bed coater. So quite a bit of force applied on these mini tabs through the coating process, and they have to be able to hold up. We can't have reduction in size by attrition during fluid bed coating. So when we took the wet granulated formulations and ran dissolution in 900 mils of HCl with SLS, all formulations showed incomplete release, surprisingly, that we attributed to, based on visual observation, some localized high density regions, like on the edges of the belly band, that much of the tablet disintegrates and then subsequently dissolves, but there are portions of the tablet that are very high density that fail to disintegrate and were very slow to dissolve. So based on this observation, we thought we've got the right process and wet granulation for managing this high drug load formulation with a highly compressible API, but there was still some fine tuning that needed to be done to ensure that we get sufficient porosity throughout the tablet to facilitate complete drug release. So with that, we worked very closely with Zach and his team at Via to in discussing ways that we could optimize the formulation based on our observations through direct compression, dry granulation, wet granulation. So our collaboration led to a series of experiments modifying the formulation. And I've got a snapshot of just a few of these now, but primarily moving away from simply using just ProSolve MCC to looking at other compositions for the binder. So looking at these three permutations that we picked here, Avosil, to show the audience here, was MCC as we had before, but incorporating perlitol, as well as MCC and Imcompress, and then finally eliminating MCC altogether and looking simply at mannitol plus starch. And really what the aim was, we need to overcome the compressibility of the API by adding an excipient in the external phase that provides a bit more elasticity, so that after compression, we have sufficient porosity throughout the tablet to allow media to permeate, the tablets to disintegrate, and dissolution to be complete. So this was the goal here. Let's adjust the external phase and see if we can overcome some of that compressibility to achieve all the targets for compression while also maintaining target performance. So when we looked at these different formulations by disintegration, so our control lot 21, you can see there was some extensive, some longer. The one that contained MCC and perlitol, their disintegration was slightly longer relative to the formulation that contained mannitol and Incompress, but ultimately the formulation with starch and mannitol also showed excellent disintegration. So the disintegration of lots 7 and 10 were significantly reduced relative to our wet granulation controls. So we took these formulations as leads into dissolution studies. And interestingly, what we found was that the MCC and Incompress formulation, although it had excellent disintegration, again, failed to fully release the drug within the test duration. However, the mannitol plus starch formulation was able to completely release. So once again, here the observation was that with MCC and Incompress, while we were getting complete disintegration, there was still some localized high density regions where the porosity wasn't sufficient to break these granules up into primary particles and drive dissolution. Whereas with starch in the formulation, there sufficient elasticity was imparted that we avoided these high density, low porosity regions of the mini tabs and were able to achieve complete dissolution. So with this result, the mannitol plus starch formulation was selected as the final formulation. So once again, the final step after producing mini tabs was to functionally coat them. Once again, the clofazimine has, it's red, as Zach said, it was used as a dye prior to becoming a therapeutic. So it's mouth staining as well as taste. So taste masking is essential, as we all know, to having a successful pediatric formulation. So prior to initiating with us, Zach and the team had titrated that coating level to find the optimum, as determined by release testing in simulated saliva, and as well as drug release in fasted simulated gastrointestinal or gastric fluid. So the key was that no drug release within 2 minutes in simulated saliva. I mean, we had making sure that the functional coat was sound enough to avoid any drug release in the oral cavity. So you see at 13% weight gain, there was still some release of clofazimine. At 15, it's significantly reduced, and at 23, it's completely eliminated. Whereas, and then when incubated for 5 minutes in simulated gastric fluid, we get full release from these higher weight gains. So with that, the 23% weight gain was selected as the optimal weight gain for the mini tablet. So we took that weight gain and the formulation, which was the Oidrogate, coated the tablets, and that comprised the final formulation. So with that, with the formulation meeting all criteria in terms of compressibility, disintegration, dissolution, and functional coating, we were able to move into clinical trial material manufacturing. Just a quick overview of the flow chart. I've gone through all these processes, but basically it's preparation of the drug product intermediate, followed by manufacture of the mini tab, and terminating with functional coating, curing the coat, and packaging. So this next series of slides, I won't go through. There's a lot of data and figures. So it's not, the point isn't to go through all the fine points of all these figures. It's to make the point that even though we're making GMP batches and clinical trial material, we're still in a development mode. We're still in a mode where we want to learn about the properties of the formulation and the process so that we can inform later optimization, tech transfer, and ultimately commercialization. And so that's one of the key benefits in working with the Style 1 Nano, is that the system can generate a plethora of data during your GMP runs that can, on postmortem, you can evaluate this data and start to identify places where it's working well, places where there might be some soft spots we might want to consider as we go up in scale. These problems can become magnified, so let's address them early. So the Style 1 Nano, and like we see on this slide too, is capable of generating a ton of data that you can use to further improve the process, even while in a manufacturing setting. So you're producing clinical batches, but all along you're learning, you're improving it for the next phase of development. So once again, just to highlight the issue that utilizing a compaction simulator like the Style 1 Nano, not only in development, but in manufacturing, just arms you with more data to be successful and for a difficult formulation like the one we're describing now. It's an absolute essential tool for success. So with that, we'll summarize. So as Zach said, we Via and AustinPx connected on a formulation problem, got a highly compactable, low density API with significant tableting challenges that really requires a lot of expertise and some advanced tools to move forward. And on top of that, to make matters worse, we've got a very specific target product profile directed at pediatric patients in a mini tablet presentation that has very little room for error to be successful. So our partnership, working together towards developing that formulation and that process, was going to be essential to the success of the project. So we at AustinPharmaceutics brought some advanced tools to the table, as well as our own deep expertise in developing drug products for clinical trials and beyond. And we were able to collaborate with Via to find a solution and a path forward for this product. Ultimately, after going through several iterations of from direct compression to dry granulation, wet granulation, we were able to find a process that worked, and with some formulation modifications, we were able to balance the compactability challenges with mechanical strength and performance requirements to arrive at the successful product and clinical trial material that allows this drug to move to the next phase of clinical development. So great success. So with that, I want to acknowledge the team. That it was Zach and I have the luxury of presenting the results, but it was a team effort, and the folks listed on this slide were essential to the success. And Zach, do you also need at this point to acknowledge? You did it at the front end, but acknowledge the funding as well.
Yeah, just in case. But yes, the NIH provided a contract that was able to fund all these development services. So thanks to them as well.
All right, fantastic. So with that, that concludes the presentation. I want to thank you all for attending, and we're going to open up the floor for some questions.
Yes, well, thank you so much for that very insightful presentation. As we move into the Q&A session, I'd like to direct the audience's attention towards the handouts module within the GoToWebinar control panel, where they'll find some additional documentation related to today's presentation to download for some further reading. I'd also like to invite the audience to keep sending their questions or comments right now for this Q&A portion of the webinar. If you're attending via GoToWebinar, you can use the questions window. And for those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already gotten some questions from the audience, so we'll get ourselves started with those. The very first question that I have for you here mentions that you mentioned early on that mini tablets are gaining traction for pediatric use. Why are they such a good fit for this population, and what are the biggest development hurdles that they present?
Yeah, maybe I'll start. Well, like I said, there's been some studies in which in these younger children, they even prefer these small mini tablets as opposed to suspensions and solutions. So there's a palatability issue. I don't know how many people in the audience have small children, but trying to get them to take medicine is not always trivial. And then additionally, while we did highlight some challenges we had to overcome, it uses processes and equipment that are readily available at CDMOs like AustinPx. And it's a solid oral dosage form, so we don't always have some of the stability issues that we may have if we're using a liquid dosage form.
Yeah, that's good. From our side, we in prior history, we were at one point in our company history, a sponsor company of our own, doing 505(b)(2) product development, and we advanced an optimized form of deferrosirox, which is an iron chelator, and that molecule is used across age demographics. So for folks that have iron overload issues stemming from, say, beta thalassemia or genetic disorders, they're diagnosed very early on. So the enhanced formulation we were developing had to treat new patients, which were toddlers and infants, all the way to adults and elderly patients. So we needed to develop a drug product intermediate that could be amenable to all of those patient groups. And so developing that intermediate that could be converted into a mini tablet to treat the pediatric population, for all the reasons Zach just described, was ideal. New patients are children, and a mini tablet is ideal for those pediatrics. But also, as those patients, grow and evolve into adult populations, you can simply convert the intermediate that goes into that mini tablet into an oral tablet for adults quite readily. So I think designing a mini tablet for pediatrics that is also flexible in how it can be modified as patients increase in age and size is ideal. So that was one of our prior experiences that we brought to the table when we engaged with Via, was designing mini tablets that also have the flexibility to reach other patient demographics.
Perfect. Thank you so much for that answer. Now, we often hear about formulation challenges, but scale up is where many products stumble. And so I've got a question here wondering, how did your team ensure that your pediatric mini tablets remained manufacturable at scale?
Yeah, I think that was the sort of the core of the presentation, is utilizing advanced tools and material characterization. So working at a small scale, large problems that arise later on in development sometimes don't manifest at a small scale or only subtly. So I think having tools like the Style 1 Nano and the FT4 powder rheometer are great tools at amplifying the signal of those small issues that become large problems downstream. So having the right tools and having the expertise to interpret the results those tools generate are absolutely essential to take something like a really challenging mini tablet, develop it at a small scale, and achieve robustness at larger scales. So it really comes down to tools and expertise.
Yeah, I guess just to kind of add on to that, we, like Dave mentioned, at Via, we had made prototype mini tabs with single presses and stuff, but we weren't able to recognize the scaling difficulties, although we appreciated they may exist until we were able to provide it to AustinPx that had both those expertise and the equipment to make that data-driven approach.
Perfect, thank you. Now, I've got a question here. few questions in one. Someone's wondering, are the mini tablets dosed direct to mouth for pediatrics? And how many are you dosing? And is there a choking risk with dosing small tablets that could be inhaled by the children?
Oh, that's an interesting question, especially about the choking. I hadn't quite thought that through. So with the mini tablets that were produced at the end of the presentation, we've now back at Via Therapeutics have been doing additional testing with different medias, dispersing the mini tabs. So they can either be directly given into the mouth. They fit on a teaspoon and can be swallowed, just like, nearly like a suspension. Or they can be added to different media and also given to children.
All right, perfect. Thank you. Now, another question here is someone saying that, AustinPx has its roots as a former sponsor company. So they're wondering, how does that history influence your approach to complex pediatric projects compared to more traditional CDMOs?
Yeah, as I described, we had been down the road before of developing pediatric mini tablets for D-ferrocyrox. So there's some expertise and some know-how that directly applies to Via's problem technically. So that's an obvious answer, but sort of the less obvious, the more indirect, is that as a former sponsor, we understand all the complexities that face a biotech in taking a concept to commercial reality, in terms of not just technical development, and considering preclinical issues, clinical issues, regulatory, commercial, scale-up tech transfer. We can appreciate those, but also you're working on with a certain amount of funding, and you've got a certain amount of runway. We understand the importance of milestones and achieving milestones quickly as they impact funding events. So we understand the burn rate of a small company. We understand when an asset is sort of at the core of the company's existence, and we live that ourselves when we bring in some like Via's project. We feel the importance of that asset ourselves. We treat it like our own. We've been in their shoes. We know exactly what they're facing, and we work as an extension of their team and a partner, not just understanding the technical problem, but also the challenges that face pharma companies in total towards meeting milestones, solving difficult problems, and moving products forward through clinical development and ultimately to patients. So yeah, that understanding of our clients' challenges is just in our DNA from our prior history, and it, I think, really makes the interaction with Austin Pharmaceutics unique relative to other CDMOs. And again, not only do you get great technical advice, but there's also sort of less tangible, indirect advice about regulatory and clinical challenges that we face, that we provide that advice to our clients free of charge, just based on our own history. We've got battle scars for our own development, and we like to help our clients avoid those same scars by giving them our advice and experience from prior history.
Perfect, thank you. Now, you mentioned stability in distribution in low resource regions in your presentation. Someone's wondering, how did you ensure that this formulation would be viable without cold chain infrastructure?
Well, yeah, I mean, I guess that really was part of the box going in. Some of those constraints that we placed on the development and the rationale for choosing mini tablets in this case for moving forward. I mean, additionally, not all the data was shown here, but there was engineering batches produced that were also placed on stability that were reflective of the zone 4. I think we did 4A, and then here at VIA we also looked at zone 4B ICH conditions. So we're and then that was also repeated on the GMP batches. So we're cognizant that that is something we have to pay attention to, and so it's something we're looking at throughout the development process to make sure that we're not going to run into that problem after it's too late.
Thank you very much. Now, the clofazimine formulation presented had some unique constraints, from bitterness to staining to mechanical fragility. How did your team prioritize trade-offs when multiple product attributes were in tension?
Yeah, I think that was a challenge. Again, kind of woven throughout the presentation. You've gotten some issues that you got multiple issues that are you've got to balance. Solving one amplifies the other. So once again, I keep stressing the same point. It's like having the right tools to identify those issues, their underlying causes, and finding a compromise for both that allows you to achieve the target product performance is essential. So yeah, it's very difficult when dealing with a formulation problem where solving one increases the other. So that's where the right tools, the right expertise come into play. Like I say, not only changing the process as we did from what we originally targeted as direct compression to incorporating a granulation phase that significantly improved a lot of the initial issues, but then as we saw with wet granulation led to some other incomplete dissolution issues. So for that, we've solved one problem and now we've created another. We need to work together to modify the formulation to maintain the solution we have to the one problem and solve the dissolution issue. So we were able to do that working with Zach and the team by taking the information we already knew, finding a formulation alternative that would allow us to solve both. So it is a difficult balance, but like I said, with the right tools, the right expertise, and good collaboration, we're able to solve all the issues and achieve the ultimate deliverables on the project and move the product forward.
Now, another question here is wondering, is the small size a risk for overdose in accidental ingestion by children?
Well, I mean, yes, it could be if they got access to it, which is why it really, or we felt like it really was best combined with maybe this counting device for this particular indication and patient population, so that we could easily adjust that while still maintaining the tamper-proof child safety constraints that the bottle provides. Also, just kind of specific for TB, a lot of times these are dosed in clinics under direct observational therapy to make sure that the dose is delivered. So while it's not maybe as particularly high of an issue for our clofazimine mini tablets, it is something to consider for mini tablets and part of the design that you want to think of. But I think this could also be an issue with just about any product. But yes, we've made them like little nerds that look like candy. Yeah.
Perfect. Thank you so much for that answer and for all of the answers today, but we have reached the end of the Q&A portion for our webinar. If we couldn't attend to your questions, know that the team at Austin Pharmaceutics will follow up with you, or if you have some further questions, you can direct them to the contact information that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also use to share with your colleagues so that they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and then gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks, everyone.
Thanks, everyone.